Well, welcome back, everyone. I hope you had a wonderful break. We're going to continue with our afternoon sessions with a presentation by Dr. Eric Green, the director of the National Human Genome Research Institute. I'm just going to give a brief introduction and then we'll let Dr. Green begin. So Dr. Eric Green is the director of the National Human Genome Research Institute at the National Institutes of Health. He is the third NHGRI director, having been appointed by the NIH director, Dr. Francis Collins, in 2009. Dr. Green's relationship with the Institute began long before his appointment as director. He served as the Institute's scientific director from 2002 to 2009, chief of the NHGRI Genome Technology Branch from 1996 to 2009, and the founding director of the NIH Intramural Sequencing Center, in which he served as the director from 1997 to 2009. Prior to that, he played an integral role in the Human Genome Project, and he has authored or co-authored more than 370 scientific publications. And so with that, I will turn it over to Dr. Green.
Well, thank you, Carla. Let me start by welcoming all of you to this year's short course. We're delighted to be putting this on, as we've done many times before. We're thrilled that you are joining us, and the you includes, I know, alumni who have benefited from the course in the past and are now helping us in various ways putting it together this year. Obviously, I would be enjoying this immensely more if we were all in the same room and doing this in person. Unfortunately, obviously, the circumstance we find ourselves in force us to a Zoom-based interaction, so we will make the best of it. But I do miss not having a chance to meet all of you in person. Because of it, I normally would make sure that all of you were given a hard copy of information about the Institute by way of this brochure. So I did want to point out, and I give you a URL, you can find it, or we could give the PDF and put it on a site that you could download it. We do have a brochure that really very much talks about a number of aspects of the Institute. It also has some really nice graphics in it that you might want to grab for some of your teaching purposes. But I did want to point you to that as a way to at least welcome you through a brochure to the Institute and tell you a little bit, by reading it, you can learn a little bit more about the Institute overall. The other thing I wanted to bring to your attention, especially because I know there's some high school teachers in particular who are taking this and thinking about using some of these materials for teaching their high school students, is a couple of years ago now, I was asked unfortunately it didn't show up. It's not showing up for some strange reason, but maybe there's basically an article in the American Biology Teacher where I was asked to put a perspective, which actually was in the form of a letter I wrote to my then graduating from high school senior daughter that talked about genomics and the journey of genomics as it paralleled her life now as a graduating high schooler. And actually what I talk about in there very much will parallel some of the things I'm actually going to talk about in this talk. And if you get this article, and we're going to make it available to you and you can actually see it, you will be able to use it if you want with to share it with students, because it was written for high school students, and it might be something that you would find would synergize with some of the things you might want to use in teaching about genomics. So with that as a backdrop, what I want to do in this talk is really cover two very broad areas, and this is going to very much be historical reflection in some ways about the field of genomics at a very high level, but with a little bit of looking towards the future. This is all about sort of the journey of this field of genomics, which has had several phases and has just built up in its excitement in every phase, and that's including the present time as we look forward. The other thing I'm going to do during this talk is I'm going to go through this first area of just talking about the first a little over 3 decades of this field of genomics, and then I'm actually going to stop and take some questions. So please start putting questions in the chat box anytime. I don't want to go to the very end and have questions. I'd rather be able to answer some of your questions halfway through. And then I'll do the second part of my talk and take more questions at the end. So one of the things to keep in mind in thinking about the field of genomics as being a relatively young scientific area is the fact that there's such a logic associated with its origins and a logic associated with its early development up to the present time. And really at its core, the fundamental basics of genomics really relate to our desire to fundamentally understand the blueprint necessary for life, whether it be life in terms of humans or life in terms of any life forms. In the case of humans, we have known for decades and decades that of the roughly 30 to 40 trillion cells in the human body, they all operate using the same fundamental blueprint. And of course, for many years, we didn't even know what that blueprint was, nor did we know what kind of molecule that blueprint was composed of. But slowly but surely, we learned a lot about that blueprint. Eventually, we figured out that blueprint resided in the nucleus of cells, and we eventually figured out these were things in structures called chromosomes. And of course, it's the chromosomes that then are sort of responsible for carrying that blueprint molecule from cell to cell, from generation to generation. But for a long time, it wasn't really known what that information molecule was, what that blueprint molecule was that was necessary for developing a living system and operating a living system. But eventually we figured it out and figured out that it was, of course, the molecule DNA. And a lot was known about DNA for a long time without really understanding how it was that it served as a fundamental information molecule, a blueprint molecule of living systems. And the key last piece of the puzzle that sort of allowed us to understand how it was that DNA could function as the key information molecule was the discovery in 1953 by Watson and Crick using the data generated by Rosalind Franklin that revealed the double helical structure of DNA. And in many ways, we could almost say that the origins of genomics really date back to something probably around 1953, because once we understood the structure of DNA, we knew, therefore, that this was going to be the key molecule to comprehensively understand if we were going to really get a comprehensive understanding of biology. And of course, it was that double helical structure with the two strands and how one's complementary to the other that revealed how it was that DNA could convey information from cell to cell, from generation to generation. And so starting in the 1950s, then we could layer on top of that fundamental information about how it was that DNA was the information molecule. Some of the earliest clues about this came in the 1960s when we elucidated through a series of brilliant studies the famous lookup genetic code, or the lookup table that probably many of you teach in your introductory biology or AP biology classes in high school, or any biology class tends to feature the genetic code, because it reflects beginning to understand how it was that DNA's letters convey in a code form information for building proteins, and through the codons that encode for individual amino acids that are then used to build polypeptides. So that was a key understanding of one way that DNA confers information in the form of genes, and how it is that genes are then used to code out information about proteins. It was then in the 1970s, and in particular the 1980s, that technologies really started to take over as we got better and better methods for cloning DNA, for manipulating DNA, and ultimately getting better and better at reading out DNA's building blocks. And of course, that's known as DNA sequencing. And in the 70s and 80s in particular, we were incrementally improving these fundamental tools and technologies we had for studying DNA in the laboratory. And one, of course, thing that's important that all of you fully understand, but it's always important to emphasize, is the simplicity of DNA's alphabet, with basically consisting of 4 building blocks they reflect 4 different chemicals that we abbreviate GATC. The complexity of DNA, of course, comes in the form that it is the order of those building blocks across very large stretches of DNA that encode all the information necessary for making proteins through genes and for regulating those genes and for choreographing many other things associated with biology. As we got better at sequencing DNA, it started to raise the notion of, well, maybe we could actually once and for all read out all the DNA of an organism. And all the DNA of an organism is called a genome. And that gave rise to the idea of, maybe we could develop a big enough program to actually read out all of the DNA letters in the human genome. And of course, while that is a big number, it is not an infinite number. And in fact, the human genome consists of about 3 billion bases, or 3 billion letters, or 3 billion nucleotides. And the idea was, could we get to the point of having efficient enough approaches for sequencing and scale it up well enough that we could actually contemplate reading out the entire genome sequence of an organism like humans and figure out the order of 3 billion letters. This all coalesced throughout the latter part of the 1980s. And the idea of being comprehensive in the study of an organism's genomes gave rise to the phrase and the field of genomics. And let me really emphasize, the word genomics did not exist anywhere in the scientific or medical press until 1987. And in 1987, with a lot of discussion about being comprehensive in sequencing the DNA of organisms and sequencing their genomes, gave rise to the naming of a new field and the creating of a new journal. And in fact, it was in the lead editorial of this new journal called Genomics that the editors described this new discipline and this new name and this new journal describing this field of genomics, and really putting out there into the scientific press the idea that there's this new field. And so that was about 33 years ago. So it's only been about 3 decades that we have had this field of genomics named as a field. And of course, the real reason why the field was named and there was all this enthusiasm in 1987 was that was nearing the end of an intense planning and strategic planning ideas and debates and arguments and actually some controversy, but ultimately gave rise to the launching of this audacious project that was planned in the late 1980s and then launched in October of 1990 called the Human Genome Project. Now, the Human Genome Project I'm very familiar with because I was actually just had graduated medical school and was then doing postdoctoral research as part of my residency in a laboratory that was heavily involved in trying to develop plans for the Human Genome Project, and in fact got funded to participate in the Human Genome Project on day one. And as a postdoctoral fellow, I was there on the front line of the Human Genome Project, working on it, actually starting at the beginning and finishing it to the end. And this project was audacious in scope and scale. It's not what biologists typically do. Typically, individual investigators working at individual labs, but this was not about that. This was about getting hundreds and hundreds of investigators working together and then all pursuing a singular project with the goal, ultimately, in the Human Genome Project of reading out all 3 billion letters of the human genome. This was large-scale biology, which was at that time fairly unprecedented, and it was also international in its design and in its organization. And importantly, it also broke down a lot of barriers along the way, including around sharing data long before you publish the data. The idea of the Genome Project was get the data out to scientists and get it out on the internet so that everybody could be benefiting from it. The project was complicated, and the project certainly had a lot of twists and turns, but the project at the end of the day was successful. In fact, instead of going 15 years as originally planned, the project was completed in 13 years and essentially achieved all of its goals. And I would point out that this is a celebratory year, and in fact, this October, we will be celebrating the 30th anniversary of the launch of the Human Genome Project. I can tell you it's hard for me to fathom that was 3 decades ago. I feel like it was maybe not yesterday, but maybe a decade ago, but it was actually 3 decades ago that there we were starting the Genome Project. The gun went off and we took off and never looked back, and then finished the project 13 years later. So I thought it would be useful with that as an early part of the journey of genomics. And in many ways, that was sort of the first phase of genomics. The first phase was very much reflected by the Human Genome Project. But now we have 3 decades into this new field of genomics, and one way I like to organize sort of a description of the main highlights is by sort of listing in a historical way what I think have been the most important contributions in genomics to date. So I'm going to sort of do that now by setting up a number of other topics. The first highlight, actually, I already told you about. I'm going to give you 6 highlights total, but the first one really will always be the first highlight, is that here we actually read out the human blueprint. We sequenced the very first human genome for the very first time, and that was done by the Human Genome Project. And that will always be highlight number 1. But that was 17 years ago, or, I mean, that was now, back in, finished in 2003, 17 years ago. What then happened in genomics was actually very interesting, almost immediately starting 17 years ago, because genomics was laser focused on the goals of the Human Genome Project up until 2003. But once the Genome Project ended, all these groups that were getting involved in genomics started to disperse the technologies of genomics to other groups, and lots of opportunities came for applying genomics to other areas. Were applied to agriculture and ancestry and livestock, infectious disease, forensics, bioenergy, microbiome. And later on in this course, you're going to be hearing from Sean Conlon, who's going to tell you a lot about microbiome research enabled by genomics, but also evolution, population history, and so forth. The point is, I'm actually not going to talk about any of these areas. You'll hear about at least one or maybe a couple of them in other talks, but it really became a very wide and expansive set of applications that genomics found its way into. What our institute, the National Human Genome Research Institute, really stayed focused on was applications of genomics to health and disease and medicine, and in part because it reflected being the lead institute at the National Institutes of Health that was focused on genomics. It became very clear that the reason we did the Human Genome Project was to set up a circumstance where we could think about how to use genomics as part of medicine in an effort to try to improve the health of humankind. And so 17 years ago, when the Genome Project ended, it was rather obvious that this area needed to be one of the major emphasis areas in genomics going forward. When I say obvious, it wasn't just us thinking about it here at NIH. I mean, I'm talking about the popular press, such as the New York Times, or the scientific press, such as Science Magazine. They recognize that the reason we did the Genome Project was to try to apply this to medicine. And so they juxtaposed those two words and started talking about genomic medicine. And it was very clear that this was going to be a key aspect of the second phase of genomics. Having completed the Genome Project, the next phase was to try to figure out how was it that we were going to enable the use of genomics in the practice of medicine. But let me be really candid. 17 years ago, it was blurry what that meant. Yeah, we can imagine using genomics and practicing medicine, but it was not well in focus by any means. That was what we needed to do, is to bring it into focus. First thing we needed to do is fundamentally understand, what did we mean by genomic medicine? And what I would tell you is, by genomic medicine, we mean truly a discipline of medicine, whereby we don't treat patients based on the average presentation of patients, but rather we begin to use information about each patient's genome, which is unique, and using that information in some way to tailor their medical care and their clinical care. And really the idea is to begin, we've always appreciated that everybody has a slightly different blueprint, everybody has a slightly different genome, but we didn't have the tools to be able to discern differences between different patients' blueprints or genomes until now. And so the idea was, could you start to use information about genomic differences among patients to tailor their medical care? And that would be genomic medicine. Now, that's a definition, but how you operationalize that is not trivial. It was going to be a journey, and that journey was going to begin with the end of the Human Genome Project. And we recognized that the finish line of this next phase of the journey was going to be using genomic information routinely in medicine. Having a cursory idea of what we wanted to do, but not necessarily knowing all the steps that were going to get us there, we recognized this was going to have to be a high priority, even to think about defining those steps and figuring out how they were going to be accomplished. I think we appreciated when the Genome Project ended 17 years ago that this was not going to be done by a single scientist or a single clinician. It was not going to be done by a single country. It was actually not even going to be done by a single discipline. And it was also not going to be done fast. It wasn't going to be a sprint to get there, but rather the analogy would probably be much more akin to a marathon. Many, many people running for a long time, shoulder to shoulder, with these different people having different disciplines. some very basic scientists, some computer scientists, some engineers, some clinicians, some nurses, some genetic counselors, some ethicists, et cetera, et cetera, et cetera. And so I think it was this deep appreciation that we were in this for the long haul. It was going to require a lot of large-scale, team-oriented science to figure this out. But ultimately, the aspiration of changing the practice of medicine was quite compelling. And we should be able to do this if we're thoughtful and sort of have a strategic approach on how to get there. And so that was what we attempted to do. And so what has transpired in those 17 years in that marathon? How far have we come? What have been the steps? What have been the major milestones on this journey? So let me tell you about some of those milestones. And I'll tell you about the next one in greater detail than the others, but I think you'll gain an appreciation for how these sort of nicely and very logically fit together. I would tell you that the most significant development since the end of the Human Genome Project, so if you said what's the number one major development in genomics since the end of the Human Genome Project, no question it would be around the development of new technologies for sequencing DNA. Because those technologies have come on board now and they have changed everything. And in fact, the highlight is truly remarkable because in the past 17 years, we have reduced the cost of sequencing a human genome by 1 million-fold. Just think about it, 1 million-fold reduction in the cost. Now, to fully appreciate why that was so important to have accomplished, let me remind you that 17 years ago, when we finished sequencing that first human genome, we celebrated, but we also recognized that it was not a cheap thing to get done. And in fact, generating that first human genome sequence by the Human Genome Project cost about $1 billion. It was well worth the money, but it cost $1 billion. So 17 years ago, one of the things we said in thinking about a high priority area for genomics was that we need to reduce the cost. I mean, $1 million per genome is way too expensive. And we need to make this more like a clinical test. We need to make this like a CAT scan or like some expensive laboratory test that you might have to get. And those are usually, maybe $1,000 or something like that. So we need to figure out how to reduce the cost of sequencing to produce what affectionately got referred to as the $1,000 genome. Could we go from $1 billion to $1,000? And how long would it take to get there? The reason it cost $1 billion was the way we sequenced the human genome the first time. We did it in factories like this. This is one of the major centers that was used in the Human Genome Project, but it involved these machines that were about the size of small refrigerators. Every one of those machines was a little over $500,000. Facilities would have hundreds of these machines. They would operate them 24 hours a day, 7 days a week, and we were actively sequencing human DNA for about 6 to 8 years. So this was really expensive. And it also took lots and lots of people constantly feeding samples into these machines and lots of computer people or computational folks analyzing all the data coming out of these machines. It was what was needed for the very first time we were doing something, but this was never going to get us to a $1,000 genome. It was going to be way too expensive being done the way we did it. We needed some fancy new science fiction-like device that was going to change everything. And the good news is that we were able to stimulate the development of that through grants and that we put out, but also the private sector got very active in this space, recognizing the tremendous business opportunities in genome sequencing, such that they came in and helped commercialize some of the early developed technologies that were coming out of our early efforts about 17 years ago, 15 years ago, 12 years ago, and so forth. And the good news is, I'll just give you the punchline, is we're there. And there's various articles that have been written in various websites, including websites at genome.gov, our website, that chronicles this. But the bottom line is we have gone from having a genome sequence of all of human DNA costing $1 billion to now readily being able to do it for less than $1,000. And that has been just remarkable. And it's not just the cost of sequencing, it's also the way sequencing is done and the speed at which it can be done. So I mentioned to you earlier that first human genome sequence by the Genome Project cost $1 billion, and it took 6 to 8 years, hardly a clinical test, 6 to 8 years of active sequencing. But nowadays we have great new technologies. We don't need big washing machine or a big refrigerator or washing machine sized machines. Now we have things that like podium size that would fit on this table right here. And with those instruments, they have little devices that you put in there, and they could read out a human genome sequence for about $1,000 or less and do it in about a day or two. And if you really speed it up, you can get it in less than a day. And so that's what's even available routinely now around the world. And in fact, I know that you will be getting a virtual tour later this week at this NIH Intramural Sequencing Center that, as you heard in the introduction, I founded and directed until I became the Institute Director. And my colleagues there are quite excited about giving you a virtual tour. And they will show you some of these technologies that are now being used that can sequence a human genome for $1,000 in a day or two. So you'll learn more about those technologies. They may also introduce you to some of the up-and-coming technologies. And some of these are pretty way cool. The one that you're going to hear a lot about from Adam Filippi, who's going to give a talk later in this course, who's one of our scientists within NHGRI, is this thing called nanopore sequencing. I don't want to steal his thunder, so I'm just going to show you this, but not describe it. But it's a very fancy technology that's very miniaturized and really, really way cool and way powerful. The remarkable thing about nanopore sequencers, and you may see this on the tour, maybe Adam will tell you, is that the laboratory requirements are minimal. I mean, you don't even need a big machine. And in fact, the machine actually consists of a device like I have right here in my hand, where all the laboratory exists on this little chip that comes out. And here you can just sort of see it. And I think they're going to show you other ones. But this is a machine. It's not a machine, it's a lab. It's everything you need. And you put a DNA sample in here, and all you need is a computer. In fact, you could even use a laptop computer. And this is a USB device that just plugs into the USB port of a laptop computer, and off you go. And then you can sequence genomes, including sequence human chromosomes that Adam will be telling you about. What's remarkable about that is it gives great portability to your ability to sequence genomes, so that you can go into the field and do genome sequencing. Shown here is a real photograph during one of the Ebola outbreaks in Africa, when they were typing the Ebola virus by reading out its blueprint, in essence, and being able to track which strains of Ebola were found in different villages by doing sequencing right there in the field with minimal infrastructure. Again, just a laptop computer. Similarly, this was done in Antarctica, sequencing some DNA from some environmental samples. And the first time ever that DNA was sequenced in space was using this exact device, using this astronaut scientist, and she was able to demonstrate you could sequence DNA in space. And so what's incredible is that the technologies are only getting better. This nanopore device is made by a company that claims that they're going to be putting out, although I haven't seen it yet, putting out a device that attaches to your smartphone. So I would not be surprised if sometime in the future we'll be able to sequence DNA on our smartphones with a little adapter such as that shown here. So you'll hear more about this, but I really wanted to unpack this highlight, because the notion that in 17 years we reduced the cost of science by 1 million-fold meant that we have now enabled genome sequencing to be routinely used at very high throughput for research applications, and increasingly now we're being able to use it in clinical applications as well. So with that advance in technology, it allowed us to now begin to think about what were the other steps that we're going to need to be accomplished to move us closer and closer to the realization of genomic medicine. And one of the next ones that with these new technologies came, the next step was going to be, all right, we now have had the ability to sequence one human genome, but we need to sequence many human genomes because we want to catalog differences among people's genomes. And just by back of the envelope numbers, as you just keep this in mind, that any two humans differ about 1 out of 1,000 places in their genome. So across their billions and billions of base pairs, any two people differ about 1 out of 1,000, which gives them about 3 to 5 million differences between any two people. And most of those differences are actually fairly frequent in the population, and that a few percent of all people will have any different spelling that any other person has. And so that becomes, again, a finite problem. If you sequence enough people, you can develop a catalog of the most common spelling differences, in other words, variations or variants in our genome, and have that information available to scientists to study. And so what has happened is we've gone from having a sequence of one human genome to now having sequence of hundreds of thousands, and probably getting very close to having millions of human genomes have been sequenced. And this has been done in a number of major projects in very organized ways that have allowed catalogs of the most common variants to be made available on the internet for scientists to be able to study. And the reason that is important is while any one of us contains about 3 to 5 million letter differences in our genome, the great majority of those differences are not biologically consequential. In other words, they don't influence our health or well-being. It's only a small subset of those that are biologically meaningful and only a subset of those that are medically relevant. And so the reason we need an inventory of the most common variants that are out there is we need to get a handle on which ones are biologically important, and even more important, which ones are medically important, because those will be a very small subset of all the variants. And we want to set up a circumstance whereby when we sequence a human's genome, we will know from interpreting it which variants they have that are medically relevant and have that be used by their healthcare professionals in developing a management plan and treatment plans for those individuals. So we have gone from having information about maybe 10,000 places in the human genome that we know people differ to now having over 100 million places in the human genome. We know people differ there, we know what their differences are, and those data are available to scientists around the world. Of course, if you have a spelling difference, if you have a variant in a stretch of DNA, as I mentioned earlier, you want to know, is that going to affect the function of the human genome? Is it going to be biologically consequential, or is it going to be totally silent? In order to be able to interpret that, you need to understand how the human genome works. And what I would say is we've spent the last 17 years working very hard to understand how the human genome works, and we've made profound advances in understanding how the human genome functions, although we clearly have a long way to go. When you really look at our progress to date has mostly been about understanding genes that encode for proteins. Since the Genome Project ended, we've now developed a very accurate inventory of the roughly 20,000 genes that are in the human genome. And so we know that part of how the human genome works. Turns out that genes only correspond to about 1.5% of the letters of the human genome. And in other words, only 1.5% of all the letters in the human genome directly encode proteins. So a small fraction of our genome is dedicated to encoding proteins that are protein coding genes. And so a larger amount of our genome that is functional, and a much larger fraction than 1.5% is functionally important, are doing things other than directly coding for proteins. Much of those sequences are involved in choreographing when and where and how much our genes are turned on. It sort of relates to the ideas of why is it all of our different cells in our body all carry the same blueprint, but different cells in our body use the genes in different ways. I mean, brain cells use one set of genes, liver cells use a slightly different set of genes, and muscle cells use a slightly different set of genes after that. And how does it, they all have the genes there in their DNA, but only through the use of these other parts of the genome that choreograph which genes are used by brain cells, which subset of genes are used by muscle cells, which subset of genes are used by liver cells, and so forth. That's where we get our complexity. And so we have begun to understand the non-gene functional parts of our genome and how they work, but believe me, we are a very, very long way to go. So if you sort of ask me 17 years after the Genome Project ended, how far have we come in understanding the function of the human genome? I'd say we've come quite a long ways, but I'm sure we're not even 10% of the way there. I bet there's 90% more biological complexity to be teased out in how the human genome works. I often have said that, I think this is going to require decades and decades of effort, probably another 2 generations before we fully understand all the complexities of the human genome. It's one of these areas that we'll just have to keep chipping away on to fully interpret all 3 billion letters and understand all the intricacies of all the different ways the genome confers function. However, we've learned enough about genome function to be able to begin to understand how certain variants can negatively affect genome function. And we've had better and better tools for being able to study that, so that what we've been able to do is begin to apply these advances in genomics technologically, and understanding about how our genomes vary and understanding about how variation affects genome function, to begin to understand how it is that genomics influences human disease. And this gets us closer to genomic medicine by being able to understand how genomic differences in some patients make them at risk for getting diseases. Now, our greatest advances so far in this arena have been with rare diseases, because rare diseases tend to be relatively simple because it's almost always a single gene that is mutated that ends up giving you risk for getting that disease. So if you think about rare diseases, you think about diseases like sickle cell and cystic fibrosis and Huntington's disease, where it's one gene that has a variant in it, a mutation that breaks the gene, and then you get the disease. And we've made, so it's simple to figure that out because you're just looking for basically a single mutation that breaks a single gene. And that's why we've made great progress. So to give you a number, when the Genome Project started now 30 years ago, there were only a handful, maybe like 61 or so, rare diseases for which we knew what gene was mutated, about 61. But today, that number is closer to 5,000. So now there's 5,000 or so rare diseases for which we now know what gene is mutated in that disease. So that's been great progress. Where the progress remains to be made, and what we will be looking for the next decade or two, are not so much rare diseases, because rare diseases have the advantage of being simple because you're looking for defects in a single gene. But more common diseases, which affect humankind much more, and by more common disease, think about the big diseases like diabetes and hypertension and cancer and cardiovascular disease and mental illness and autism and Alzheimer's and so on and so forth. Those are very common, but it turns out they're much more complicated because it's not a single gene that has a variant in it, but rather it's multiple genes and what is typically a greater contribution of other factors like the environment and social context and so forth. And so here we have much more progress that needs to be made in understanding the genomic basis of common diseases. But here we are quite empowered with new technologies, better and better understanding of variation, better understanding of genome function, that I think we will see greater progress in these areas of understanding the complexities of common diseases in the coming decade or two. So those are the 5 highlights I in particular always like to stress that have really we've seen in genomics. But what's particularly gratifying is over the past 5, 6, 7 years, I've been able to add another highlight to this that really reflects some of the earliest recognition that we're going to reach our destination eventually in realizing genomic medicine. Because what we have seen come about really, maybe in the past 6, 7 years, has been some vivid examples of genomic medicine emerging. And I really want to stress, these are just the early examples. I think there's going to be many more. They're sort of like tip of the iceberg, but I think it's reflective of and illustrative of what we are likely going to see going forward as more and more opportunities come for implementing genomics in medicine. And I'm going to have to be brief about this in light of time, but I did really want to stress that we've gone from having a very blurry view of what genomic medicine might look like to now actually seeing it, at least for some early examples, being very much in focus. And so I'm only going to give this in highlight form. I'm frequently asked, what are some of the earliest implementation of genomics in medicine? What are the hot areas? And there really are 4, I think, that we can speak about. And I would point you to reading more about this through some Google searches. But one of the hottest areas in clinical genomics would definitely be in cancer. Cancer is a disease of the genome, and the reason you get cancer is you have cells that pick up many genomic changes. And we can read out the genome of a tumor as effectively as we can read out a sequence of a normal cell. And we can, now it's being operationalized in many, many ways to improve the management and the treatment of cancer. So cancer genomics is probably the hottest area of genomic applications in medicine. Another area you're going to hear more and more about is just a big word, pharmacogenomics. It's just pharmacology and genomics. Why is it some medicines work well in some people and not in others? It's because we have slight differences in our genomes that influence drug metabolism and the pathways that we use to metabolize and deal with medications that we take. And we're learning more and more that we can predict people being either a good responder or a bad responder to a medication by first getting genomic information about them and being able to get clues, so that instead of just randomly picking the best hypertensive medication to give somebody, we could be more rational by first doing a genomic test and then selecting of the available hypertensive medications which ones to give. And so pharmacogenomics is absolutely standard of care for some medications, and the hope and expectation is that list will grow. Another area which gets a lot of popular press attention is the idea of having patients with undiagnosed conditions or very rare conditions where once upon a time there was really no way to figure out what was wrong with them. But now, literally, you sequence their genome and you can figure it out about half the time. And this isn't just done once or twice a month nowadays. This is done literally dozens and probably hundreds of times every single month worldwide. Patients are getting their genome sequenced and diagnoses are being made. This was unimaginable 17 years ago, the idea that you could actually diagnose a disease based on a complete genome sequence, and now it's become quite routine for patients with rare diseases. Once again, it's when they have relatively simple single genes that are mutated, being able to figure this out. Again, routinely done hundreds of times a month around the world. And lastly, there's a test that's actually now done probably 50 to 60 million times a year worldwide, involves prenatal genomic testing. And now prenatal testing has been around for a long time, but not the way it's being done now. Though it's been around for a long time, where it involves having to access fetal DNA through procedures like amniocentesis to be able to basically use a needle to access the fetal DNA. But now these new methods for sequencing DNA are so sensitive that they allow you to access fetal DNA that is naturally shed from the placenta that floats around in the maternal bloodstream. So through a simple blood draw of a pregnant mom, you can use these new technologies and some very fancy computational work to basically determine whether there's any routine chromosome abnormality in an unborn child, again, through a non-invasive test, a simple blood draw. And so that is why literally 50 to 60 million pregnant women will get this genomic test done this year. #1 use of genomic medicine to date. So I'm going to pause there, because that now covered the first part of my talk. The second part is much shorter. And I just want to pause and just ask Carla or Christina or somebody, are there any burning questions? Maybe I'll take 2 or 3 questions and then resume.
Okay. So I'll start off with one, Eric. So Mary Scott, she pointed out that, there has been a decrease of, you said, 1 million-fold for sequencing genomes. And she asked, can we hope to see the same reduction in costs occur in gene therapy?
So it's a great question. And in fact, gene therapy and even some genomic-enabled drug therapies and so forth that are so expensive, we don't know. So, but taking specifically gene therapy, there absolutely are some major research projects that are going on. Some of them funded by NIH, some of them funded by the Gates Foundation, the famous Bill Gates, that foundation. And actually, there's even one program where we're jointly funding it to try to develop methods for gene therapy that would allow, and it gets pretty complicated pretty quick, but it would not have to involve hospitalization, would not have to involve very complicated and very expensive ways of delivering the things that are needed to make gene therapy happen. And the reason that's needed is that for worldwide applications, especially in parts of less developed parts of the world, it's just not practical to do gene therapy if you don't get the cost down, and the cost is going to have to come down by having it be not having to require hospitalization and so forth. So absolutely, there are efforts to develop very fancy technologies that maybe one day would reduce the cost of things like gene therapy, and it would be truly seismic if any of these work out. Another question, Carla?
Sorry, Eric, I was muted. I was talking to you, but you couldn't hear me. Another question from Andrew Lee. Other than omnipresent threats to funding, what do you envision as the greatest obstacles to continue delivering the promise of genomics? For example, less data sharing, public privacy concerns, distrust of science in general, technology consolidation.
That's an excellent list. So I think that's an excellent list, but I would add one more, because it probably would have been my first answer. And when you say the promise of genomics, in particular, promise of genomics for in medicine, I would actually say the #1 threat to this is the imperfection of healthcare. The imperfection of healthcare in the United States, the imperfection of healthcare every place in the world. Everybody has have good systems and bad systems. And the reason I'm stressing it is I'm not sure this is unique to genomics. I mean, I'm a physician, I can say this. I mean, I think healthcare in America, it's wonderful and it's horrible all at the same time. I mean, we have miraculous capabilities, and it's basically a dysfunctional system and how it gets paid for and how it's organized and the disparities that inherently get introduced. So I think that's the greatest threat. It's actually not unique to genomics per se, because we have the same problem with medications and with access to technologies and so on and so forth. So I think if we could solve healthcare, I think genomic implementation would go smoother, but right, as you pointed out, maybe not perfectly smooth, because there will be concerns about distrust, there will be concerns about discrimination, there will be other ethical concerns come in. But I actually think the number one threat to it is just the generally challenging, dysfunctional nature of healthcare delivery in the US and actually in many other places around the world as well. I'll take one more caller and then I'll
okay. So one more. So, and this is just from your folks here at ECIB. This year, we received more applications from middle school teachers than ever before. As genomics starts to be introduced earlier in classrooms, what do you think could hook kids to go into something genomics related?
No, that's a great question. And I guess I've sort of watched this in various ways, and Carla knows this because she's helped me in some cases. I've been, as my kids were growing up, I got pulled into teaching in some of their classes or interacting with some of their science teachers and so on and so forth. And I have absolutely would agree with the idea that I believe middle school is the time where you can start getting people interested. I think you, who am I to tell your assembled teachers how to, because that's their trade. But so I'm not sure I know. I'm only speaking maybe as a parent. It just seems to me that there are ways in particular of making genomics interesting and fun. We try to do some of this at the Institute. But, I think it is one of those areas that you can use various hooks, whether it's you want to do it from the forensic side or from the ancestry side or from the disease side or from the, that I think can get the hooks in to then wanting to understand the fundamentals. I don't think you need, probably not start with the fundamentals, but start with what I think can be some really cool and fun applications of genomics that really, and even things like, fur color, or eye color, or hair, lots of simple traits. There are those things that certainly we've been involved in at the Institute. Carla knows far better than me, thinking about how to develop materials for different age groups. But I absolutely think middle schoolers are ready for this, and it would be great to get them interested and engaged then, and then have a build on it through high school and beyond. So those are great questions. And we'll take more at the end. Let me move to the second area, which really it's not going to be nearly as long by any means, but I often find that it's important to pause about this point in my talk because if you didn't happen to notice, I'm very enthusiastic and I make it seem like all this stuff is just crazy simple and it's all went so well and it was all so easy and it's all incredible. But I like to shift gears and point out that I'm actually also a realist, and that there really are some tough things we're constantly dealing with in this journey of genomics. Now, there come with some opportunities, but I always like to share with groups like this group some of these realities, because I do think it is important to recognize that there are a number of things that we are still working on, a work in progress. And I have a number of these I often talk about. I'm just going to share 3 of them with you. So reality and opportunity number 1 is that I've probably slightly misled you into thinking that reading out somebody's genome and interpreting is simple, straightforward, and it's just a slam dunk, and we do it all the time. The truth is we do the first half of it all the time, and it is reasonably simple and straightforward. We could generate a human genome sequence pretty readily. My colleagues at our sequencing center and their virtual tour will convince you of this. I can tell you, we can read out somebody's genome for less than $1,000 in like a day or 2. However, we can even put it through a computer program, and we can even read out and determine the 3 to 5 million spelling differences, the 3 to 5 million variants in that person's genome. But if that person is a patient, and that patient has a disease or some sort of health ailment, and we have those 3 to 5 million variants, and we round down that patient the next day, I can tell you that this is what we still feel like. we have lists and lists and lists of variants, but we don't always know what, well, in fact, we never know what all those variants mean. And if the patient has some complicated or even a common disease, we really can do very little with that sequence at the moment. The bulk of our knowledge is actually, how to use this clinically is actually quite small. So in other words, clinically understanding that genome sequence is not trivial, even though we can generate it. So we have a gap between what we technically can do and what we can technically understand. And while we've made great strides, we're really working hard to reduce that gap. So a huge priority for us is to get better and better at understanding what all these variants mean. We know what some of them mean medically, but we have a long way to go. So don't, just believe me, we are still working on this very, very hard. A second thing that I think becomes, maybe it's even relevant to what we were just talking about, how to get younger people interested in this, is that, you know, once upon a time, I don't think most people thought genomics was very interested. It really wasn't really relevant to them. I mean, when I got involved in the field, it was just a field relevant to some geeky scientists like me that were, you know, working away on the Human Genome Project. It was not relevant to people beyond that. It was just something for a small group of scientists trying to map and sequence the human genome. Now, 17 years ago, when the Genome Project ended, yeah, we got healthcare professionals interested in working with us because they came into the tent and they started to see that genomic information might be useful for the practice of medicine. But, you know, besides that group and us, it was still not quite relevant to everybody. But things have started to change now, because now all of a sudden, patients with cancer, genomics becomes relevant. Patients taking medications where pharmacogenomic information is relevant, then genomics becomes relevant. Families and friends and patients and the patients themselves with rare diseases who get their genome sequenced, wow, genomics is relevant to them. And what about all those pregnant couples, the 60 million of them who are getting the prenatal genetic testing done now with this new genomic test? Now genomics is relevant to them. Once you touch healthcare, as genomics is doing now, genomics becomes relevant to patients and their friends and their relatives. And so now all of a sudden, instead of being some small little discipline off in the corner, genomics is now relevant to everybody, and genomics is now relevant to society. And so this becomes something that we are very cognizant of, that raises a whole host of ethical issues and social challenges and legal implications and so on and so forth. And some of it could be the hook to get people interested in it. But, you know, thinking about some of the big questions about ethics around genomics, whether it be related to health disparities, whether it be related to the use of genomics in forensics, the use of genomics in terms of preventing discrimination and so on and so forth. There are many, many issues that are relevant at the interface of genomics and society. And some of those could become tools for teaching students about genomics and the broader implications of it into medicine, but also into society. So a reality and an opportunity. Another reality and opportunity, and this one is a fun one, it's the last one I'm going to tell you about, is the recognition, and some people think that by this point in my talk, I think genomics is the only explanation for human disease, but of course it's not. I fully appreciate that for most diseases, hypertension, diabetes, cardiovascular, all the diseases I named earlier, it's a constellation of factors that contribute to the risk for the disease. Some of them are spelling differences in our genome, but others, such as shown in this pie chart, are related to aspects of our lifestyle, our environment that we live in, our social context, and so forth. The reason you've heard a lot about genomics and its role in health and disease is because we've had this incredible technological revolution that have allowed us to dissect the left side of this pie chart. But there's a similar revolution of technology for dissecting the right side of this pie chart, because we are getting better and better, not me, not this institute, but other scientists and other technologies being developed elsewhere for measuring better and better the environment and aspects of our daily life. And what that means is that there are now technologies that allow us to collect immense amounts of data about people, not just their genomes, but about their physiology and their life and what they're exposed to. And there's all sorts of review articles, just one that talks about some of these way cool technologies that really are the moral equivalent of the new technologies for DNA sequencing. This is now new technologies for monitoring other aspects of our life. And what that is setting up is opportunity for a new field that goes beyond genomics and genomic medicine into something that is now being frequently referred to as precision medicine. And this is sort of the new journey, if you will, the next phase. It's even going slightly beyond genomics. And precision medicine is just simply a more precise accounting of individual variability. Everything up until now in my talk, I've talked about individual variability in our genomes, our genomics. But I'm talking about more precise accounting for individual variability in our physiology or in our lifestyle, how much we exercise, how much we sleep, what we're exposed to, and things that we do to our body, like what we eat or what we do in terms of things like sun exposure or unhealthy habits like cigarette smoking. And we're getting more technologies for measuring lifestyle and environment and physiology. And that is setting up a circumstance whereby we can begin to holistically look at all different aspects of people's lives at a very large scale in terms of data. We can take genomic data about people and we can marry it with data that we acquire through these new technologies, how much people, how well they're sleeping, how much they're exercising, what they're exposed to, et cetera, et cetera. Oh, and by the way, while we're at it, since we're analyzing lots of this big data, let's throw in it the fact that 15, 20 years ago, we really didn't have access to a lot of data that people collected when they got medical care. But nowadays we do, because everybody has electronic health records, and increasingly those records are being made available for scientists to be able to study. And so it's this confluence of genomic advances and new technologies with the availability of electronic health record data that has led to this new field of precision medicine. And like the Human Genome Project, a very large project has been established here in the United States called the All of Us Research Program. There are similar projects in other countries. In fact, some even started before the one here in the United States. But this is sort of the modern version of the Human Genome Project, but now going beyond the genome. The All of Us Research Program is a program in the United States that is ongoing, that is actively recruiting 1 million or more people who are going to participate in a study that will be followed for probably several decades. These people who are participating will share their genomic information. They're going to also share biological samples, and they're going to share their electronic health records. And all this data is going to be collected and secured, but then made available for researchers around the world, including citizen scientists and probably high school students, to be able to come and analyze that data and be able to tease out various aspects of people's health, their well-being, the practice of medicine, and so on and so forth. And we'll be able to get the full pie chart of the contributions to health and disease. If you want to read more about this, I strongly encourage you, maybe even want to sign up and be a participant. But to me, this would be a very exciting thing to share with your students. They could follow it, and there'll be lots and lots of ways of engaging with this program, either as a participant, but also following them with a lot of the things that they have associated with it, educational tools, and even being able to access some of their data. And so this is a brand new area, is a huge opportunity for research where we will go beyond understanding how the G's, A's, T's, and C's influence our health and well-being. We're going to increasingly get a better idea about how other aspects of our life influence our health and well-being, and how that information can be used to tailor our medical care so that we are not just practicing genomic medicine, we'll be practicing precision medicine. And finally, I just want to stress before I close that I also think it's scientific to convey to students is that all this stuff is not simple. And while you read the high-level summaries of this, it sounds like all this happened so easy and seamlessly. The Genome Project, the development of the $1,000 genome, the operationalization of genomic medicine. None of it was linear. It all was a journey of very many twists and turns and surprises. And that's the fun part of science. It's actually the fun part of scientific inquiry, is that it's the challenge of being able to adapt and constantly facing things that require you to think about new approaches to try to accomplish something. In my office here at NIH, literally just around the corner from where I'm sitting in my conference room, above my office door, I have a quote from this guy who I hope you all recognize, because I think it really much captures the spirit of genomics, where Einstein said, if we knew what we were doing, it wouldn't be called research. And believe me, no aspect of these different phases of genomics, every one of those phases of the journey, we didn't really know what we were doing. That's why it was research. We were constantly adapting, twisting, and turning. We're doing it at the present time. It's what makes it exhilarating. It's what makes it compelling, and what makes it a very rewarding career. No matter what aspect of genomics you're participating in, you always have challenges, and you're always adapting to unique circumstances. So I will stop there, but just close by saying I'm happy to stay connected with you and your students or anybody else. You can stay connected either by subscribing to a monthly newsletter that I put out called the Genomics Landscape. One email, if you want a month, please feel free to sign up for that, because I often give updates about the kinds of things that I talked about. And you can hear about this from me and my staff. We put this together once a month. And anybody who's interested to follow me on Twitter, it's another way I try to get messages out from the Institute and from things going on in genomics. So with that, maybe I'll leave this slide up instead of going to the question slide and turn it back over to Carla. And I know I'm at the hour, but I'm happy to take another few minutes of questions if there are questions you want me to answer.
Okay, well, thank you, Eric. I want to say, again, thank you so much. I do want to give Belén the opportunity to ask you a question, which I think will sort of bring this together very nicely. Christina asked you the great question about middle school students and how we get them connected, but Belén had a cool question personally about you and your perspective on where we are. So go ahead, Belén.
Well, I was just kind of wondering, because you said it's been 30 years from the beginning of the Genome Project, and I was there at the beginning. And I was wondering if you could back in time and you had 5 minutes to spend with yourself 30 years ago, and you probably had some ideas about where things were going to go, and some things didn't take off, some things were way beyond what you ever thought. So if you had like 5 minutes to whisper something in your own ear 30 years ago, what would be the highlight that you would tell to yourself, or like, don't go there, or this is going to be amazing, something that pops in your mind?
Maybe, well, it depends. If I was going to go back and talk to myself 30 years ago, the revelation I have, which I would have probably got in an argument with my 30-year-ago self, because I wouldn't have believed it 30 years ago, is if I go back and tell myself 30 years ago, that you will, so because the other little subtlety here is I was just out of medical school, I was training clinically in pathology, and my attraction to genomics was my belief that genomics might one day in the diagnostic arena have significant influences on the practice of medicine. But 30 years ago, I was always thinking, wow, I could see that happening like in the next generation, if my generation is successful in making genomics a reality. I would not have believed 30 years ago that during my career, I would have started to see genomic medicine implemented. It just, and the idea of 30 years ago, if you would have told me, while you are still an active in the field, while you are, have a job in genomics, we will be routinely sequencing a patient's genome and making diagnoses of rare diseases, I would have said, no way. I mean, that just would have been completely not even contemplated. I mean, it just seems so science fiction. It would be the kind of thing that, yeah, maybe one day, but that has got to be a generation or two off. And here we are 30 years later. I still think of myself as young. I'm still in my career and we're doing it. So I would think that's what I would tell them. And I think what I would probably also tell myself is there's going to be a lot of blood, sweat, and tears that you're going to endure. During the Genome Project, there were some moments. And even beyond, none of it's going to be easy, but it is absolutely going to be worth it. You really are, the genomics is really going to move the needle in medicine. So, persevere and convince people this is worthwhile.
Thank you.
Wonderful. But thank you, Eric. I think that's the perfect way to wind up your presentation. Thank you so much. If it's okay, if we have any other questions, we'll make sure we get them to you. But I can see people giving you hands claps on their little icons there. Thumbs up and hand claps, since we can't see you in person.
Right. All right, well, enjoy the rest of the course. You're in good hands with this crew and the others that are going to be involved. And so enjoy yourself. We're delighted to having this, and please make the most of it. That's what we want. We want you to get the most out of it.